and let me talk about the beta-arrestin technologies. The very first arrestin technology was essentially, it was looking at a translocation of arrestin in cells to the surface of the cell when the cell was activated. And I think it was a U2OS cell was the cells they used. They used those because they were flat and round. It was a direct measurement of arrestin translocation, but the arrestin had to be tagged with a green fluorescent protein. The GPCR was not tagged, but you had to have the GPCRs in really high expression level on the surfaces. It did require imaging. It was very low throughput, but it was the very first way that we really could look at arrestin movement. The other thing you could look at is ERK phosphorylation. However, that is a downstream signaling event. It's not as proximal to the receptor as you'd like to see. Assays also have been developed for enzyme complementation. Most of the enzyme complementation assays required tagging of both the arrestin and the receptor with complementary pieces of the enzymes, and that actually works. That actually works fairly well, but the concern is that tagging of the GPCR itself may alter the pharmacology of that GPCR, and that may be a concern when you're really trying to make the best interpretation of the data. So there are opportunities for the development of an ideal HTS arrestin assay. The ideal assay would require no tagging of the receptor. That way there should be eliminate the potential artifact from a large tag. You should be able to read it in a plate reader without imaging. You should be able, it would be a direct measure of translocation or activation. And the ideal assay would measure all signal transduction in one cell. So both arrestin and G protein mediated. So that way it would avoid issues pertaining to differences in receptor arrestin expression from cell type to cell type. It would avoid differences in other internal signaling ingredients basically in that cell, which also would complicate interpretation if you do it in two different cells. So that's why if you measure everything in the same cell, you can compare apples to apples versus apples to oranges. So the ideal assay would also be amenable to primary cells. So we have developed, in collaboration with some folks at Columbia, a multi-screen beta-arrestin technology. And this is what the technology basically is based on. So essentially, we have an arrestin, and the arrestin is tagged with the N terminus of a luciferase. And there is a C terminus of the luciferase, which has a membrane locator sequence. So it basically gets stuck in the membrane. So these two, the arrestin and the luciferases, when they come together, it stimulates the luciferase being active. So essentially, when a ligand comes and activates the GPCR, the arrestin moves to the GPCR. The luciferase is complemented by the luciferase in the membrane, and then you get a signal. So you can study signaling bias in a stable cell line or primary cells with this assay. It's a true pharmacology in vitro and in vivo, no GPCR tagging, and it doesn't require any GRK co-transfection as well. So this is actually what we get with looking at the mu-opioid receptor with a transient transfection. This is with DAMGO, the ligand for it. And what we see essentially is when you add a lot of our ligands or a lot of the arrestin constructs, you wind up with no signal. Just there's a sweet spot where you add enough, you get a really nice signal transduction of arrestin movement. So a good luminescence. The protocol is really, really easy and very amenable to transfer from one person to the other. Essentially, you seed cells in 384 well plates, you incubate overnight, you wash with PBS, add substrate, incubate for 5 minutes, treat with compounds for 20 minutes, and then you read luminescence. And then you analyze however you want to analyze the data. We use GraphPad Prism. The total assay time is only 30 minutes. We have here the mu-opioid and kappa-opioid assays in a stable cell line, and this is basically the time course. This is 10, 20, and 40 minutes. So this is a 40 minutes 10-minute. This is the ratio of signal to background. And you can essentially see that with your ratio of signal to background, it's actually very, very stable through the 40 minutes. It may be stable longer than the 40 minutes, but this is how stable this is for this particular time course. The CHO K1, this is the 1 minute. 1 minute, this is 10 minutes. And here is your ratio of the kappa opioid receptor in the CHO K1 cells. Essentially, you have a very, very stable assay. And this is the delta opioid and nociceptin stable cell lines, and your time courses with that as well. So from 1 minute to 40 minutes, and here is your ratio. So essentially, you can find a nice time point where you can read the assay where it's going to be stable throughout and be able to run it on your 384 well plates in high throughput and have enough window to be able to do that. Here with the nociceptin receptor, you can see that the 1 minute was not adequate, but everything else was. Now, one of the questions was, does having a stable cell line with the beta-arrestin signaling, alter your cyclic AMP signaling in those receptors? And so these are basically the same things we've looked at. So mu, kappa, and nociceptin receptors, basically looking at what happens to the EC50 of those receptors in the arrestin stable cell lines. And essentially, the EC50 we get for this is identical to the EC50s we get with non-transfected cell lines that don't have the beta-arrestin constructs. This is the beta-arrestin assay in the stable cell line again. So this is with mu opioid receptor. This is the different ligands, and you can see really nice dose responses. Here is dose response for a dynorphin. This is dose response for this ligand, and dose response here. Again, this is the stable clones, different cell numbers, and you can look at your different responses. So essentially, this is for beta-2 adrenergic receptor, vasopressin receptor, and the cannabinoid CB2 receptor. So essentially, we've moved not just on the opioid receptor families, but we've moved it into other receptors, so beta-adrenergic, vasopressin, and the cannabinoid receptors. I said before that we can do all the assays within a single cell background. And so this is a mu opioid receptor and all of the different assays in that same cell. So cyclic AMP, this is our internalization. We can measure phospho-ERK. We can look at arrestin. Here's our arrestin signaling with our arrestin constructs. This is GTP gamma S. We also do saturation binding and competition binding assays. So we can, essentially the idea would be that you could run something like this and be able to compare within the same cell what happens with your compounds from compound to compound to compound with an unambiguous determination of exactly how those compounds behave and what the actual signaling bias is. We've developed several cell lines now with this, and I've told you about all the ones basically. So mu, kappa, delta, nociceptin, beta-2, and the V1A receptor. These are the different assays we have in those cells, and here's the beta-arrestin, the new one. So how do you get access to this? We can develop custom cell lines for you. So we would generate stable cell lines with the multiscreen sensor, select and optimize in the arrestin assay. So it would be, essentially, it would have the sensor and your GPCR of interest, and that would be a 3-month turnaround. Also, you could license the technology from us. We would transfer the sensor and the protocols to the client, and then the client, you guys can develop the cell lines and assays yourself. So summary, basically, we have developed beta-arrestin sensors to study all signaling bias in one multi-screen stable cell line or in primary cells. It's a true pharmacology in vitro and in vivo without GPCR tagging. No GRK-TOE transfection is necessary. It enables ligand-induced beta-arrestin detection in a native and high-throughput manner. Simple protocol and assay setup with a total assay time of 30 minutes. We found actually that we can transfer this assay from person to person in the lab without any kind of issues. It's a simple transfer. The assay is really easy to run.